There are two sorts of reactions in photosynthesis, the light-dependent reactions and the light-independent reactions, and you've probably already figured out the difference between those two, so that's nice. The light-independent reactions are called the Calvin cycle. Yes, that Calvin cycle. Photosynthesis is basically respiration in reverse, and we've already covered respiration, so maybe you should just go watch that video backwards. Or you could keep watching this one. Either way, I've already talked about what photosynthesis needs in order to work: water, carbon dioxide, and sunlight. So how do they get those things? First, water. Let's assume that we're talking about a vascular plant here. That's the kind of plant that has pipe-like tissues that conduct water, minerals, and other materials to different parts of the plant. These are like trees and grasses and flowering plants. In this case, the roots of the plants absorb the water and bring it to the leaves through tissues called xylem. Carbon dioxide gets in and oxygen gets out through tiny pores in the leaves called stomata. It's actually surprisingly important that plants keep oxygen levels low inside of their leaves for reasons that we will get into later. And finally, individual photons from the sun are absorbed in the plant by a pigment called chlorophyll.
In the aquarium, plants are responsible for performing several actions which are beneficial to the overall water quality and in turn our fish. In order to accomplish nutrient uptake, oxygen production, and carbon fixation, plants first have to harness and convert the energy coming from the light above. This process is called the light-dependent reaction, which happens only during the photoperiod or the time in which your aquarium lights are on. Plant cells located on the epithelial layer of leaves can contain tens, if not a couple hundred, small organelles called chloroplasts. They've been determined to be evolutionarily related to cyanobacteria, which is pretty cool. Inside of each chloroplast is yet another structure called the thylakoid. It's another membrane-enclosed system that houses all the machinery necessary for the first step in photosynthesis. Let's take a journey closer to the thylakoid membrane where we can get a better understanding of how the light reaction works. Embedded within the membrane of the thylakoid are two distinct photosystems that work simultaneously, but with their own agenda. Both photosystem 2 and 1 house chlorophyll A and B molecules whose job it is to harvest and pass light energy to a special reaction center, chlorophyll. Now the type of available light here is important. Both chlorophylls absorb light in the blue and red portion of the photosynthetically active range that falls within the visible light spectrum. This photosynthetically active radiation, or PAR light, spans from about 400 to 700 nanometers, and again the chlorophyll found in plants will primarily absorb the blue and red light present in that spectrum. This is why full spectrum and or 6500K bulbs are most common on planted tanks. Now we'll talk more about PAR and bulb types in another video, so let's get back to the light reaction. Let's start here with photosystem 2 that uses light to excite a pair of electrons that are passed on to an electron transport chain. Water molecules are split into free hydrogen ions and oxygen, effectively replacing those electrons, continuing the cycle and creating the essential proton gradient or proton motive force across the membrane of the thylakoid. Now because the protons on the inside will be in a higher concentration than outside, they will naturally diffuse out, passing through a molecular machine called ATP synthase, which converts ADP into ATP, the energy currency of the cell. Now those electrons that went into the transport chain are passed through the membrane by various carrier proteins ending up at photosystem 1. Light is used here yet again by chlorophyll molecules as a way of re-exciting those electrons so that they can be utilized in reducing NADP+ into NADPH, a compound that plays a vital role in many biosynthetic processes, including carbon fixation. At the end of the process, the plant has generated ATP, NADPH, and oxygen, all from the power of light energy. The oxygen is released into the environment and is also shuttled down the plant to the roots, where it's used to prevent necrosis that would otherwise take place in the anaerobic substrate. NADPH and ATP will be used by the plant in the Calvin cycle, where CO2 is converted into sugars, which ends up driving cellular respiration and even more ATP generation, as well as carbon for assimilation. This will allow the plant to grow and function the way we want in our aquarium. Now, optimizing the lighting can be really important in your planted aquarium because without the energy produced as a result of the light reaction, your plants won't be able to assimilate as much carbon and will therefore consume less of the leftover nutrients that cause algae problems. Now, of course, the other half of the photosynthesis equation is often more crucial in the home aquarium, so next time we'll go over the Calvin cycle in detail and discuss why having available CO2 can be extremely effective in the heavily planted aquarium.